Hello biochemistry class, welcome to chapter 4. In this chapter we're going to learn a little bit more details about amino acids. So this chapter we're going to break it up into two different parts. The first part is going to be talking more about amino acid structure. We're going to talk about the general structure of an amino acid, as well as how the stereochemistry works there, and then we're going to discuss the side chains of each of the physiologically relevant amino acids. In the next lecture, we're going to talk more about the reactions that the amino acids can undergo. So to start out, we'll talk just about the basic amino acid structure. So as you should already be familiar with, amino acids have Amino acids have an amino group as well as a carboxylic acid, which is partially where they get their name from. Those are both attached to a central carbon, and this carbon is chiral because it's attached, with one exception, to 4 different groups. So one group amino acid, one carboxylic acid, one hydrogen, and then the last group is an R group, which we'll usually refer to as the side chain. And this structure on the left is shown completely uncharged, which is a common way to sort of generally draw an amino acid, but it's important to keep in mind that at different pHs, the amino group and the carboxylic acid group will gain and lose protons based on the pKa's of those groups, as we've discussed a little bit before. And in fact, there is no pH at which you would actually see the structure on the left where both of these groups are uncharged. So at physiological pH, we see more often the structure that we see on the right, where our amino group is charged and our carboxyl group is also charged. The other thing to mention, like I said, is that the chiral carbon in the center is chiral. So it's important to think about the arrangement of the groups around that chiral carbon. And this drawing is actually a Fisher projection. And so with a Fisher projection, you can always make the assumption that the horizontally drawn bonds are actually wedges, so they're coming towards you out of the page. And the bonds that are drawn vertically are actually dashed lines, so these bonds are pointing away from you. And this helps give you hopefully a better idea of what a three-dimensional amino acid would look like based on the orientation around that alpha carbon. So let's talk a little bit more about that stereochemistry. So all amino acids have a chiral center with the exception of one, and we'll see more about that when we discuss the side chains a couple slides from now. And all of the standard amino acids in proteins are in the L configuration. And this L configuration refers to their similarity to the arrangement of atoms around L-glyceraldehyde. So that's where we get that L naming convention. It's actually not directly related to how light is rotated, which you may have also learned about in organic chemistry with levorotary and dextrorotary. The amino acids are actually split in terms of the way they rotate light, depending on which side chain they have as that R group. But it is important to know that in terms of configuration of atoms around the chiral center, they are all in the L configuration. And additionally, you probably also learned about S and R notation for the chiral carbon, or the chiral atom in the center. And so we can also note that all of the amino acids, with one exception, except cysteine, are in the S configuration. None of them are in an R configuration about that chiral carbon in the center. So it helps you to see here the way this is named. This is an L amino acid with that alpha carbon being the chiral carbon. And something that you might have noticed is that the way that this amino acid is drawn on this slide is a little bit different from the way the amino acid was drawn on the previous slide. So they both have the amino group on the left side as a wedge coming towards you, but actually the carboxyl group on the previous slide was also drawn as the wedge coming towards you. And here it's been rotated up one, and so it's now pointing away from you. And also the R and the proton have switched places. And this doesn't actually mean we pulled apart any of those bonds and made a rearrangement around the chiral carbon. This drawing is also correct. And they're both the same. They're showing an amino acid in that S or L configuration. And really what happened here was that the carbon, this bond between the carbon and the amino group rotated. So all of these other three bonds sort of rotated around one and kind of swap positions. And that's what we see here, because there's free rotation about all of those bonds, but the configuration of the way those bonds are arranged will always be like how we see here and also here, because they're actually the same. And so if any of you still have a modeling kit that you may have had when you were learning organic chemistry, this might be a good time to pull that out and sort of try to replicate the structure of an amino acid and rotate around all those bonds and see, hopefully that will help you get a better sense of how those are the same. So that being said, let's move on to talk more about each of the side chains that we're going to be seeing that are important in a biological context. So first we have our non-polar side chains. And I just want to note that on all of these slides we're discussing the side chains. Part of the amino acid is drawn in black. That's the part that doesn't change. That contains the amino group, the carboxylate group, and the hydrogen. Then there's a part that's drawn in red. That's the R group, or the side chain. And they're drawn in red here just to help them really point out themselves to you. So I will make my pen maybe a green color so it sticks out. So our first amino acid that we have here is our simplest one, it's glycine, and the R group is a hydrogen. So like we hinted at before, this is our one non-chiral amino acid, because now our central alpha carbon has two bonds that are to the same group. So this is our one exception with our non-chiral alpha carbon. The next biggest side chain in our nonpolar group is alanine. It's got just a methyl group thrown on there. And as you'll see, most of these nonpolar side chains are mostly going to be hydrocarbons. Carbon and hydrogen don't have a big electronegativity difference between them, and so these bonds are considered nonpolar. So the different amounts of carbons and hydrogens that you see will define the different side chains, and we'll see at the end of this list that there are a couple of other atoms thrown in there as well. The next one that we have is valine. It's got 3 carbons in this sort of V shape. That's how I remember the structure of valine. It's got a V shape, just like the name. And then for leucine, we have that same valine V shape, but with the addition of an extra carbon there in the middle. And then we have isoleucine. So leucine and isoleucine, you might have noticed, have very similar names, and that's because they contain actually the same essential number of carbons and hydrogens, just arranged differently around each other. And what makes isoleucine special, so just sorry to make that more clear, between leucine and isoleucine, essentially you've moved this sort of bridging carbon over to this side of the V, so one side of the V is longer. But what makes isoleucine more interesting is it actually has another chiral carbon because of the way the atoms are arranged. So this is our second chiral center, which is drawn here with a star. Moving on from there, we now have methionine. Something that makes methionine interesting is it actually has a sulfur in that hydrocarbon chain. And we often think of sulfur actually as more of a polar molecule. It has a little bit more electronegativity to it than a standard hydrocarbon bond, but since it's in this hydrocarbon chain, we consider methionine as a whole, that side chain, to be nonpolar still. So that brings us to proline. Proline is special, and you definitely probably already noticed this because it looks very different. It's special because the side chain actually is a ring structure that attaches both to that alpha carbon as well as to the nitrogen that's in the amino group. So here in the middle, you see our alpha carbon, and that side chain with the 3 hydrocarbons comes off of there and also attaches to the amino group that is part of the regular standard amino acid chain. And this gives proline some special properties that help give proteins a certain shape, essentially because proline is being forced into being a very particular angle with its bonds because we added an extra ring in there between the alpha carbon and the amino group. So next we have phenylalanine. Phenylalanine is aromatic. It's got an aromatic ring structure on the end here, which is still all hydrocarbons, so we consider it nonpolar. And then last we have tryptophan, which has another aromatic ring structure to it. And this one contains a nitrogen, which you probably noticed. And a lot of times we do think of nitrogens as being more polar molecules, but because on the whole, this side chain is primarily hydrocarbon, we also just consider this overall to be a non-polar side chain. And now that we've sort of seen a little bit of the structural differences, I want to go into some of the naming conventions. So every amino acid that's biologically relevant has a name, a full name that we see written at the top on each of these. I'll use glycine as our example. So this amino acid is called glycine. Each amino acid also has two different amino acids. They have, two different abbreviations. So the first abbreviation is a 3-letter abbreviation. So for glycine, it's gly, the first 3 letters of the name. And that's true for a lot of amino acids. It's often the first 3 letters, but not always. And I'll point that out as we go. And then also all of the amino acids have a 1-letter code or abbreviation. And again, that's often the first letter of the amino acid's name, but not always. So I'll go into some exceptions. So some exceptions for the 3-letter naming convention, we have our isoleucine. So the 3-letter abbreviation isn't ISO, it's ILE. And then for the 1-letter abbreviations, we see also something you might not expect for phenylalanine. The one letter code for phenylalanine is F. And that's essentially because you'd expect it to be P, right? Except P is already taken because proline is P. So you have to be very careful when you're referring to the amino acids by their abbreviations to make sure you get the one you are intending to get. If you write P, make sure you're talking about proline. And if you want to talk about phenylalanine, make sure you refer to it as F. And similarly for tryptophan, the abbreviation for tryptophan, the 3-letter code is TRP, not TRY like you might expect. And its 1-letter code is W because another amino acid that we'll go over shortly already has the T. So keep that in mind as you're learning the names and structures of these amino acids. So the next group of amino acids that we'll discuss are the polar side chains. So these amino acids, the side chains, the bonds are polar, but they are uncharged. So they have neutral charge. So what we'll see here, as you might expect, are some oxygen molecules in these side chains, as well as some nitrogen molecules, and a couple of examples of sulfur molecules, which are sufficient enough to make the side chain as a whole considered to be polar. So we'll start at the top with serine. Serine starts with the hydrocarbon, and then it has the hydroxyl group on the end. And next we have threonine. So threonine is another special one that has an extra chiral center in it that's attached to the hydroxyl group that helps it make it polar. Next we have asparagine. So asparagine has an amide group that you might recognize. And glutamine is very similar to asparagine, except it has an additional carbon there in the side chain. But otherwise, the structures are very similar to each other. Tyrosine is our aromatic polar amino acid. So it has the aromatic ring, but in addition, it has the hydroxyl group. That's enough to help consider this whole structure as a polar structure. And then on the bottom of this list here, we have cysteine. So cysteine looks a lot like serine, right, which we saw at the top, except instead of a hydroxyl group, it has a sulfhydryl group. And the sulfhydryl group gives cysteine some really special properties that we'll talk about that are drawn on the right side of this slide. So when two cysteine residues are close to each other in space, if they're on parts of a protein that are near each other, they're actually able to join together to make a covalent bond. And this covalent bond is called a disulfide bond. And so essentially what happens is the two hydrogens from the sulfhydryl group are removed, and through an oxidation reaction, the sulfurs are joined together to make this disulfide bond. And this is actually a covalent bond. It's very strong, and it can help to give the protein a lot of stability in the structure at this location, because this disulfide bond will really hold those two amino acids to each other very closely in space. And so, like I mentioned, this reaction to create a disulfide bond is an oxidation reaction. And if you wanted to reverse this bond, to break that disulfide bond, to go back to having two sulfhydryl groups, this would be a reduction reaction. So now that we learned about our polar and uncharged amino acids, it's time to move on to our last group of amino acids. So what these amino acids all have in common is that they are charged. So some of them are positively charged, and we refer to them as basic. Some of them are negatively charged, and we refer to those as acidic, which can sometimes be a little counterintuitive for people. A good way to think about it is that the basic ones, as we talked about, bases are proton acceptors. So these bases have accepted a proton, and so they are all now positively charged. On the other hand, our acidic ones, we know that acids are proton donors. So these acids have donated their protons away, and that makes them negatively charged. So acidic side chains are negatively charged at physiological pH. Basic side chains are positively charged at physiological pH. And of course, when these side chains are charged or not charged, of course, depends on their pKa and the pH of the solution that they're in. So let's talk a little bit more about each of these charged amino acids. So starting out with our positively charged ones, we have our lysine, which is a long hydrocarbon chain that has that positively charged amine group at the end. Next, we have our arginine. So arginine has this guanidino group over here, and again, that one of them is able to be positively charged, and that gives it the basic property. These two basic groups, lysine and arginine, are almost always charged at physiological pH because their pKa's are relatively high. So and that makes them different from our last positively charged amino acid, which is histidine. So histidine's pKa is lower than the two for lysine and arginine, and that because of that, the side chain can sometimes be charged and sometimes be uncharged at physiological pH, depending on some of the other conditions. And I just want to point out here that this imidazole ring is what defines histidine, and that's where the nitrogen is that can have the positive charge to it. So, as we'll see going through practice problems and other things, you'll see sometimes that histidine side chain has a charge and sometimes it's not, as opposed to lysine and arginine. So, and again, I just want to remind everyone to be really careful with these naming conventions. So arginine, for example, the one-letter code is R, and lysine's one-letter code is K, which may be a little counterintuitive. So let's then move on to the negatively charged or acidic amino acids. So we have the two of them, aspartic acid and glutamic acid. They're very similar to each other. They both have this carboxylate group on the end. And the main difference is just the length of this hydrocarbon chain. Aspartic acid has 1, glutamic acid has 2 carbons. And we'll often refer to them by the name for the charged carboxylate group. So instead of calling it aspartic acid, sometimes you'll see this amino acid called aspartate, especially when this oxygen is negatively charged, it's lost its proton. Similarly, glutamic acid you'll sometimes see referred to as glutamate. And be very careful not to mix up glutamic acid and glutamate with one of our polar amino acids, glutamine. Those are different. Glutamine is a different amino acid, right? It has this amide group. And something also to note about glutamine, it does have a very unusual one-letter code. Its code is Q. So yeah, just keep in mind all of those things. Keeping the structures categorized in the right category can also sometimes be a little bit tricky. Like we mentioned for our nonpolar ones, depending on what source you use, maybe something like methionine or tryptophan could be considered polar because of those sulfur and nitrogens that are included. Sometimes tyrosine is even considered nonpolar because it has this big bulky aromatic group. So just be aware that the sources you use, depending on where their cutoff is, how they define polar and nonpolar, they may not exactly categorize the amino acids the way I'm showing you here. But something that doesn't ever change are things like the pKa's and the 3-letter codes and the 1-letter codes. Those are always consistent. And of course, the structures of the side chains are always consistent. So with that being said, now you should be ready to answer the practice questions about amino acid structures.